Okay, so let's move forward. So we discussed about the amino acids and how they make the peptides and then proteins. If there are more than 40 amino acids that make the protein, and then if there are less than 40 amino acids, then we call them peptides. So now we move towards another thing, which is protein. Okay. So before going to the protein, let's discuss. This is a cliche statement we heard in our high school that DNA controls all the cell activities. Do you agree with this statement?
Yeah.
So my question is how DNA controls all cell activities?
Well, it takes part in every single cell in your
Yeah.
body.
How?
Um, it's just found in every cell. That's all I.
Okay. It is found in every cell, but what does it do?
It makes proteins, I think,
Oh,
through transcription and translation of
yeah.
RNA.
So that you are coming close to it. So basically, we have DNA, right? So DNA is present in the chromosomes, okay? And each of our human cell has 46 chromosomes, and then they have a lot of DNA, right? Here is the one thing that you have to write in your notes. A particular sequence of DNA that codes for a particular protein is called gene.
Can you repeat that?
Particular sequence of DNA that codes for a particular protein is called gene.
Is a gene?
Yeah. So now what is gene? If I ask you what is gene?
It's a particular sequence of DNA that codes for a particular protein.
Yes. So it's clear or not clear?
Yeah, it's clear.
Okay. So if someone asks you what is gene, you can easily ask them that this is gene. Okay?
Hmm.
Now clear. So we have in the nucleus, we have chromosomes, right?
Hmm.
And the chromosomes have the DNA. Okay? And the particular sequence of the DNA, right, that codes for a protein is called gene. Okay? So
Hmm.
it's not denim genes, right? It is gene. It is not denim jeans that we wear. It is gene,
Hmm.
right? A bad joke. No problem. So, yeah. So it's a so I want to emphasize it again. What is gene?
A particular sequence of DNA that codes for a particular protein.
It's called gene. Okay. So for how does it do that? It does it by two processes, which is called transcription and then translation, right?
Hmm.
And then we get the protein. Okay.
Hmm.
And this process is called gene expression. Okay. If I say gene is expressed, what will be the end result?
A, a protein that's used.
Yeah. So if I say a gene is expressed, it means what is formed.
A protein.
And if I say protein synthesis, so it is actually meaning gene expression.
Okay.
So, so these are the two same things like protein synthesis and gene expression. They are same thing.
Hmm.
Okay, fine. Now, uh, there are two types of proteins, majorly, right? For the
Hmm.
sake of our work, one are called functional proteins, another are called structural proteins. Okay, so when I say the DNA controls all cell activities, so you see, We have different organelles in the cell. We have different organelles in the cell,
Mhm.
right? For example, mitochondria, for example, endoplasmic reticulum, for example, lysosomes, Golgi apparatus, centrioles, right?
Mhm.
All those things are made from, like the membrane of those things is made from what?
From DNA?
From protein.
Oh, from protein.
And that protein is formed from what?
From um
DNA.
RN. Oh.
DNA, right? So DNA is making all the, basically DNA is making proteins and those proteins are making the organelles, right?
Mhm.
Fine.
Mhm.
Then we see there are a lot of chemical reactions that are happening in the body. So these are called structural proteins, like the proteins that are making the organelles membranes, right? For example, the cell membrane, right? The cell membrane has a lot of proteins. Those proteins are formed by the expression of some particular genes, right? So those are called structural proteins. Then it's a cell. Then there are a lot of cells make the tissues, and then the tissues make the organs, right? And then organs make the organ systems, and then organ system makes the organism, right? So basically, if we start, so it is the DNA that is making the proteins, right? By this process, which is called gene expression, and then that that makes the structural proteins. And then there are chemical reactions that are happening in the body. There are a lot of chemical reactions that are happening in the body. For example, glycolysis, for example, hexose monophosphate pathway, for example, glycogenesis, glycogenolysis, protein synthesis, cholesterol synthesis, different types of processes that are happening in the cell, right?
Mhm.
And those reactions are done by the enzymes, right?
Mhm.
Those reactions are done by the enzyme.
And
So if there is no enzyme, there will be no reaction. And what are enzymes?
they're a type of protein.
They are also proteins, right?
Mhm.
So how will they be formed? If I say you, for example, there is an enzyme called pepsin,
Mhm.
right? How will how is it going to be formed?
From a
By by
uh by
by
DNA.
gene, by some gene,
Oh, I see. Okay.
right?
Yeah, yeah, yeah.
So if I say DNA controls all the cell activities, how it controls? By making proteins. That proteins either serve as a structure of the cell or that protein performs the chemical reactions that are happening in the body or in the cell. Got it?
Mhm.
Is it clear, right? So
Yeah.
all the things that are happening in, so basically it is a code. DNA is a code that is there, right? We have to appreciate, we have to understand this thing on a cellular level as well as on an organism level, that DNA controls the cell activities. This is very powerful sentence. DNA controls the cell activities. How? By gene expression. By gene expression. Okay. What is the end result of gene expression?
Protein.
Yeah.
Mhm.
What is the end result of gene expression?
Protein.
Protein. Now I take you to the pancreas. I take you to the pancreas, right? And in the pancreas, we say there is a hormone which is called insulin.
Mhm.
Okay, so insulin is produced. What is insulin?
It's an enzyme.
No.
I or
Yeah, well, it's a hormone, but what is
or
it made of? Protein.
oh.
Protein, right? So
Mhm.
insulin is a protein. Okay, so if insulin is a protein, so do you think that there will be a gene for the insulin that is there in the pancreatic DNA of the pancreatic cells,
Yeah.
right?
Mhm.
Or no?
Yes.
Okay. For example, if I talk about cyclooxygenase, what do you need by this cyclooxygenase? What is this cyclooxygenase? So whenever the word ase come to anything, it is an enzyme,
Mhm.
right? So cyclooxygenase, it is involved in inflammation. It is involved in inflammation or pain production, right? If I inhibit this enzyme, which is COX, cyclooxygenase COX, what happens?
It won't work.
Inflammation will not
It won't
happen,
express. Mhm.
right? So there is a drug which we commonly use, painkillers. So painkillers does what? They just inhibit the COX. They inhibit the cyclooxygenase. So if they inhibit the cyclooxygenase, what happens? We don't feel headache, we don't feel inflammation, right?
Mhm.
Similarly, the people who are taking cholesterol lowering medications, right? So there is an enzyme which is called HMG-CoA reductase, which is present in the liver. So I have a drug which is called statin, okay, atorvastatin or something like that, that inhibits HMG-CoA reductase,
Mhm.
right? So what is HMG-CoA reductase?
It's what produces
It's a
cholesterol.
protein. When I say ase, anything which ends in ase, right?
Mhm.
HMG-CoA reductase, cyclooxygenase, or DNase, or RNase, or protease, or lipase, all these are what? Enzymes. And by default, if they are enzymes, then it means they are proteins. And if they are proteins, so most of the, if they are proteins, then there must have been a gene for that protein. So it's a very simple thing, but very complicated it becomes when we see on a whole medicine, like whole medicine that is happening in our body, like all the chemistry that is happening in the body. So you have to understand this very simple thing. Everything in our body somehow is dependent upon proteins, right? So this is the scheme. This is not just one cell. There are trillions and trillions of cells in our body, and all the cells are doing the same work. That there is a DNA, then that DNA is transcribed to make messenger RNA, and then messenger RNA is translated to make protein. And that protein can be a structural protein or that can be a functional protein, for example, enzyme, right? And there are now millions of proteins. If there are, I mean, there are millions of proteins, and all those proteins are formed by what?
From genes.
From gene. Got it?
Mhm.
So I have to connect you to the gene therapy. That is why I am discussing all these things, right?
Mhm.
Got it? Is it clear or it is overwhelming?
It's clear.
Sure,
Yeah, it's a lot, but it's clear.
it's not a lot. It's very simple. Simple. This is a just principle. If I hear a word of protein, immediately my mind start working. Oh, there should be a gene for this protein. Okay,
Mhm.
got it? That is it. So if I say there is some protein, what you should have in your mind?
A gene for the protein.
No gene. There is a gene which made this protein. Okay. So for example, in our brain cells, there is a transporter which is called GABA, GABA, GABA. Okay. I mean this is a kind of a neurotransmitter, GABA. Okay. And then there is a chloride channel in that GABA. But what I want to say is that if I say if I say a chloride channel, so channel is actually a protein. Channel is actually a
Protein.
protein. And then we take the sleeping pills. We take the sleeping pills. Pills interact with that channel and cause us to sleep, right? So what I am trying to refer, I mentioned some drugs, for example, NSAIDs, for example, insulin, for example, HMG co-reductase, for example, GABA. Or okay, then there are blood pressure lowering medications. Okay, so we say beta blockers. We say beta blockers. Now beta receptors are proteins. Beta receptors are
Proteins.
proteins. If I say alpha receptors, what are they?
Proteins.
If I say nicotine receptor, what are they?
Also proteins.
Protein. If I say, um, for example, Janus kinase,
Enzyme. Also
what is that?
a,
Enzyme is what?
a protein.
A protein. If I say proton pump,
Also a protein.
also protein. So proton pump inhibitors are used for lowering the acid in our stomach.
Mm.
Okay, so they are called proton pump inhibitors. For example, omeprazole. So now we are clear somehow that most of our body is working because of proteins, right? And most of the all the proteins are made from the codes of gene, right?
Mm.
Clear?
Yes.
So the traditional drugs that we use, more than 95% of the traditional drugs that we use, they actually interact with proteins. They actually interact with
Proteins.
proteins. Okay, got it?
Yes.
But in our case, our target is not protein. Our target is not protein. We actually destroy the messenger RNA. If we could destroy the messenger RNA, what will happen?
If can you repeat that question?
If we can destroy the messenger RNA, if we can destroy the messenger RNA, what will happen?
Then translation won't occur to make the protein.
So there will be no protein. So there are two ways to inhibit protein. Either we inhibit the protein directly, or we destroy the messenger RNA. Okay,
Mm.
and if we destroy the messenger RNA, what will happen?
Then there won't be any protein.
So that is what is gene therapy. So basically, what we have siRNA, small interference RNA, siRNA. Si is small, RNA is capital. So siRNA destroys the messenger RNA. siRNA destroys the,
Messenger RNA.
and what will happen then?
No protein is
No
formed.
protein. So there are some faulty proteins, for example. There are some faulty proteins, some abnormal proteins that are causing disease.
Mm.
If we could destroy that messenger RNA, then those faulty proteins will not be formed. And if those faulty protein is not formed, what will happen?
Then you won't have the disease.
That's what it is. So this is our future. This is the next decade, right? So
Mm.
up till now, all the medicine that is working on protein. Okay, it is working on
Proteins.
protein. So I gave you a lot of examples of proteins. For example, proton pump, GABA receptors, uh, then HMG reductase. Then I talked about the cyclooxygenase. Then I talked about insulin. Then I talked about different, like, you name it, name any drug and go to the mechanism of action of the drug, that will translate into protein, that will go somehow to the protein, right?
Mm.
Okay, and now the problem comes when we study the science papers. We have to differentiate. When I say genus kinase, I should immediately know it's a protein. When I talk about any receptor, I should know that this is a protein. If when I talk about any ion channel, I should know that this is a protein, right? If I talk about any structure, I should know that this structure is made from protein. And then if I talk about any protein, it should come into my mind that, oh, this is a protein, so it means that there is some gene, and that gene is being expressed, and that gene should make the messenger RNA. And that if I could destroy that messenger RNA, that protein will not be formed.
Mm.
Is it clear? or is boring?
No, it's clear. I like this stuff.
You like it. So now, so so there are two terms which are very confusing, but they are same. One is gene expression, other is protein synthesis. Okay, is there any difference between gene expression and protein synthesis?
They have the same result. So,
They have the same results, right? They
mhm.
have the same results. Gene expression, protein synthesis. What is that result?
In a protein, the
A protein.
formation of a
A
protein.
protein, right? Okay. Now there is a problem. Let's see what is that problem. Okay. Let's see now. Documents. Okay. First semester. Fall 2019. Maybe fall 2020. Okay. What did you get by this? Just in a word, explain me what I said in a
Uh,
sentence.
Genes make up protein.
Oh, very good. And
Um, Gene expression and protein synthesis result in the same process. Anyway, same
and and how is cell made of? What is cell
uh,
made of?
from uh, proteins.
Proteins. Obviously, there shouldn't be any doubt, right? So most of the cells are made from what?
Proteins,
Proteins. There are two types of proteins. What are those?
functional and structural.
You see that traditional target of the most of the traditional drugs, for example, proton pump inhibitors, COX-2 inhibitors, is what?
A protein,
And the target of siRNA is what?
the mRNA.
mRNA. If we destroy the mRNA, what will happen? There will be no protein. Okay. Ah, okay. Yes. Uh, yes. And here is now Nobel Prize. Okay, there was a Nobel Prize given to a scientist on discovering this. I don't want to go into detail, but just want to say something. When we see the genome, human genome, when we see all the genes in our body, right? Which
Mhm.
was like that project was completed in year 2000. Okay, we know all the genes in the human body. Okay, but we realize that there are only certain genes like the DNA is big, there is a lot big length of the DNA, right? But there is a small portion of the DNA that codes actually for proteins. Okay, and the rest of the DNA is called non-coding region. Means we don't know what it does.
Mhm.
We know that this code, like if I have a length of the DNA and I see that this particular portion of the DNA makes the protein. Then there is empty space. Then again comes a portion of the DNA that makes a protein. Then again comes the DNA that makes the protein. But then there are those regions which make the proteins. They are called coding regions. They are called what?
Coding region.
Coding regions. And then there are non-coding regions. Coding regions because they code for proteins, right? And then there are non-coding regions. We don't know what they do. We didn't know now. We know now, but previously we didn't know that. What do they do? There are non-coding regions that we didn't know what they do, but now we know what they do. Okay. Now we go back to our births. We go back to our births, right? There was a sperm and there was an egg. What was sperm? It was a cell. What was an egg? It was an egg. It was also a cell. They combined to make what?
A baby.
Baby came after 9 months, but before that it was zygote. What was that?
A zygote.
Zygote. Zygote is a cell. Okay, and all the genome that is there in that one cell is actually us. Like all the information of the organism all the DNA that was required to that purpose, like for making an individual or making a plant or making an animal or making a human being, all that genetic material was present in what? Zygote. Okay. Now we see then, and another thing you need to know that whatever happens after forming the zygote is mitosis. Is what?
Mitosis.
And what is mitosis?
It's the cell division.
Cell division. And it is called constant cell division. Okay. It means what? That there is not going to be any change in genetic material, right?
Mhm.
But then we see uh, some. So if you understand a little bit of embryology, what happens is that after zygote we becomes morula, then there becomes three layers: ectoderm, endoderm, and mesoderm, and then ectoderm makes some organs, then mesoderm makes some organs, then endoderm makes some organs, right? But you have to appreciate that the cells of my nose have genes. They are the similar genes that were present in the zygote, because naturally, if you think the argument, everything that happened to the zygote was mitosis, and mitosis there was no change. There was no change in the genetic material. So every cell, like every cell of my body, has the same genetic material. Is it clear?
Yes.
Right? Every body, every cell has the same genetic material. Now my question is, then why some cells are behaving like something and other cells are behaving like something? Eyes are seeing, kidney is making the urine, heart is pumping the blood. They're performing different functions, but they have the same genes. How does it happen?
It's because those genes are only expressed in certain parts.
Yeah, so that is called switching on, switching off phenomenon. Switching on, switching off phenomenon. So certain genes are expressed in certain cells while remaining turned off, right? So for example, there are, just giving you an example, there are 100 genes. There are just 100 genes in a cell, right? So
Mhm.
one gene is expressed in one cell while remaining 99 will be off. Remaining 99 will be
Ah,
off, like they will be silenced.
mhm.
And then that particular gene that is turned on is actually causing that cell to behave that particular way. Got it?
Mhm.
So those non-coding regions, like those non-coding regions, later we got to know that those non-coding regions are involved in silencing those genes. Got it?
Mhm.
No. So I am showing you a human being in a very small period of time, how human beings were formed and what actually our body works like, right? So I started my story with protein. At this moment, scientist Sarah should know that scientist Sarah, whom I'm speaking to, should know that every cell is made from proteins, right? And every chemical reaction that is happening in our body is because of enzymes, and enzymes are also proteins, right? And all those proteins are formed by the expression of genes, okay? And then I wanted to tell you that there is a big chromosomes, but there are like length of the DNA, but not all the DNA that is there in our cells codes for proteins. There are some non-coding regions as well, and those non-coding regions does what? They silence the genes that are not required by some cells, that are not required by some cells. Is it overwhelming? Because I don't want to overwhelm you. I just want to act, because this is actual real thing that is what we are going to do later in our next sessions, right? So if these concepts are not clear, we cannot do biology, we cannot do chemistry, we cannot do science, right? So for understanding science, these are the big, and these are all things that were taught in high school, but not this way, right? So is it clear or not?
Yeah, it's clear.
What is clear? I just want to. I mean, is it a new thing for you, or you heard it already?
Most of it is review from the bio
Okay.
classes I
It's
yeah, but not in this. It wasn't taught in this way, but it has a similar concept to what
yeah,
I learned in bio
yeah.
classes.
So we are just connecting the dots. We are connecting mitosis. We are connecting gene expression. We are talking about transcription, translation. We are connecting all that stuff that we studied in our high school, and now we are going towards real diseases, and we are going to solve the real problems, right, in our human body. But that will happen after some like sessions. It is not going to happen right now, right? So for now, for today, I think it is enough for you to understand. If I give if you I, if I ask you, for example, in this particular case, if I ask you darosha,
Mm-hmm.
what is this darosha? A protein,
Protein.
a protein.
Mm-hmm.
If I say you DGCR8, what is that?
In enzyme or protein.
It's a protein, right? If I say exportin 5, what is this?
Protein.
If I say TRPB, TRPB, what is this?
Protein.
If I say dicer, what is this?
Protein.
If I say risk loading complex, this is made up of three proteins:
Mm-hmm.
AGO2, darosha, and DGCR8.
Mm-hmm.
If I say you RISC, RISC, RNA induced silencing complex, again, this is what? Protein. Okay, there can be very difficult names, but we are not concerned about difficult names. We are concerned about the concept that these are the proteins, these are the
Protein.
proteins.
Mm-hmm.
Okay,
Got it.
so our body is very simple, very simple. We are made from just proteins, lipids, nucleic acids, right? Proteins, lipid,
Nucleic acid.
nucleic acids, right?
Mm-hmm.
And then comes like carbohydrates. So just four things. What are those four things?
Nucleic
Proteins,
protein.
protein, carbohydrates, lipids, nucleic acids, right? Proteins, carbohydrates, lipid, nucleic acid. So your hairs are made from what?
From protein.
From proteins, right? So we say that proteins, right? So hair is made from protein, right? Skin is made from what?
From proteins.
Protein. Which protein? I mean, no, no, no. Which protein? And lipids and carbohydrates,
Okay.
right?
Mm-hmm.
Protein, lipid, carbohydrate. Protein, lipid, carbohydrate. Nothing more. Nothing is going to come extra from uh, elsewhere, except from mineral and water that also is there. But so, and there can be different scientists give very different names, very difficult names like TREP-B, DIGCR8, Darosha, I mean, RISC. So when we go to them and we say we cannot understand them, it's very easy. We have to first appreciate that if anyone could understand that what is protein, and understand that protein is made from gene, okay, and it is made by the assembling of the amino acid according to the code of that gene, right? Then they can easily understand biology, right? And I was just saying another, I gave you a very powerful statement. There are two types of DNA: coding DNA, which is called coding region, and then there are non-coding regions, right? And then those non-coding regions are responsible for silencing the genes in cells where they are not required. But those genes are there. Those genes are there. Gene of insulin is there in my eye, but it is not expressed in the eye cells. The gene of insulin is present in in my hair cells, but it is not being expressed in hair cells. The gene of insulin is present in my heart cells, but that is not being expressed in heart cells. It is only expressed in the pancreas. Got it?
Yeah.
Is it clear?
Yes.
So how it is not being expressed in eyes? Because it has been silenced, right? How it is not being expressed in heart?
It was silence.
It was silenced. How it is silenced? That will be our next topic in the our next session, so we can plan it next time. Uh, and I will explain you this diagram. What is that? I just want you for this today. I just want you to understand there is DNA, then that is transcribed to make messenger RNA, and then that is transcribed to make protein, and then that protein does all the wonders in our body. Okay.
Mhm.
Those wonders, wherever you go, whatever you see, you will find protein. You will find protein. Now let me go to the Google Scholar. Again, scholar.google.com. So we were talking about tumor targeted delivery, delivery, Meenakshi Sharma, sorry, Meenakshi Sharma. This was the last time we said it, right? Okay. See, very difficult. So, see, the oleic acid conjugated peptide showed the highest efficacy in siRNA uptake and silencing of kinesin spindle protein. So we are actually doing what? Silencing a protein which is called KSP, kinesin spindle protein, right? So previously I explained you what is CGKRK. Now I
Uh,
explained you. Now I explained you what is. What does this paper talk about? It is silencing a gene. It is silencing a.
G.
When I say it is silencing a gene, I can say that it is silencing a protein.
Can you repeat that question?
If I say it is silencing gene, can I say that it is inhibiting that protein
Yes.
or no?
Yeah, you can say that.
Huh? If I
Yeah.
say I want to silence Janus kinase, what should I do?
It.
I don't want Janus kinase to be present in the cell. What should I do?
Oh, then you target the mRNA.
That you then you that I, then first of all I will see the gene of that Janus kinase.
Oh yeah, you'll still see it there, but it's just silence.
But we have to silence it. But you are not getting my question. My question is very simple. I have a protein which is causing a lot of trouble. Name of it is Janus kinase.
Mhm.
Okay, and I don't want it to be present in the cell. What should I do?
Then you target the gene so it's not expressed.
That then I will target the gene which is making that protein.
Mhm.
Got it?
Yeah.
So we'll move forward. We will move forward and it. And for now, I think it is sufficient for today. Just to simulate it. Okay,
Mhm.
and then we will move forward. I am ending this meeting now. You have any questions, you can ask me.
Okay. Really quickly, just to clarify, so the functional proteins are the enzymes, and then
Mostly, yeah.
mostly enzymes, and then the structural proteins are the ones that make up the cells.
Yeah,
And
they make up the cells structures, they make up of cell membrane, they make the organelles membranes, they make the nuclear membrane, They make the mitochondria, they make the muscles, right?
Hmm. Okay, got it.
And what else?
Um, and then um, the non-coding regions are responsible for what silences the genes,
Why do we need to silence the genes?
so that you don't have multiple genes working in parts of the body where they shouldn't be working.
Yeah. So we want certain genes to work in certain cells.
Hmm.
We don't want every gene to work in every cell.
Right.
If it happens, then every cell will be behaving the same way,
Hmm.
right? But my eyes have different work. My hands have different work. My heart has different work. My lungs have different work. My kidneys have different works, right? My
Hmm.
hairs have a different work, right? So all that thing, what will happen? Like then we just need. I have thousands of things. I just want one thing to be expressed in one particular cell. That is why the cells are different, although they have the same genetic material. Because understanding is clear. Zygote, after that mitosis. If there is a mitosis after that, what will happen? Like there shouldn't be any change in genetic material of the cells. If every cell has the same genetic material, then why they are behaving differently? Because in some cells certain genes are on while remaining of off. And then a scientist was awarded Nobel Prize for discovering this thing, that non-coding regions are actually responsible for silencing the genes, right? And now we have the power to silence genes. We can literally silence any gene, right? We have the power to literally silence any gene. Any gene, we can silence any gene, and it is very easy. So our next classes, like our next sessions, will be about that.
Okay.
And then you will be ready to come to lab, right?
Mhm.
Are you willing to come to lab?
Yeah, I'm fully vaccinated since I'm working in the pharmacy, but I just need to block out a time to be able
Uh,
to do it this semester if I can.
okay. These lectures are different. I'm just experimenting a new thing because I have been teaching all this stuff to the like to Sean, Kelly, Jamie, but I didn't record it. So I thought I should record it so that I shouldn't be able to say it again and again and again and again, right? So I hope you will learn a lot from these sessions. If you have any questions, please ask me. And also, I would like you to re-watch this video,
Okay.
right? That will help you understand.
Okay.
So I will see you next time. Whenever you are available, just tell me, and so that we can have another session.
Okay. I'll
Okay?
let you.
All right. Thank you so much. Have a
Thank
very
you.
good day.
You too.
Bye-bye.
Bye.
Bye.